Hello. In today's video, we will learn about cell division, cell cycle which includes interphase and M-phase, mitosis and meiosis in detail, along with their significance and differences. By the end of this session, you will be having enough conceptual knowledge to help you answer questions pertaining to cell division, cell cycle, the behavior of chromosomes during karyokinesis, significance of and differences between mitosis and meiosis. We will start with cell division. All multicellular organisms start life as a single cell, the fertilized egg, and grows by addition of new cells. The development of a multicellular organism from its unicellular zygote takes place by cell division, growth, and differentiation. The division of the nucleate cells shows two different activities, namely division of the nucleus known as karyokinesis and the division of the cytoplasm known as cytokinesis. In animals and plants, there are three types of cell division: direct cell division or amitosis, equational division or mitosis, reduction division or meiosis. We will look into amitosis first. Amitosis is seen in unicellular organisms like bacteria and protozoans. It was first described by Remick in 1841 in the red blood cells of chick embryo. Amitosis is a means of asexual reproduction. During amitosis, the nucleus elongates, takes a dumbbell-shaped structure, divides into two nuclei. This is followed by the constriction of the cytoplasm, resulting into two daughter cells. But amitosis is rare. Next is the cell cycle. The cell cycle shows a series of changes or sequential events in the life cycle of a dividing cell. The period between two successive cell division is called generation time. The cell cycle includes the period from the beginning of one cell division to the beginning of next cell division. The cell cycle consists of two main phases, that is, interphase and M-phase. Look at the diagram carefully. You will see clearly that interphase is further subdivided into G1 phase, S phase, and G2 phase. Again, M phase or mitotic phase is subdivided into prophase, metaphase, anaphase, and telophase. Before going into each one in detail, let's revise what we have learnt with the help of schematic diagram. The cell cycle consists of two phases, interphase and mitotic phase. The interphase is further subdivided into G1 phase, S phase, and G2 phase. The mitotic phase includes prophase, Metaphase, Anaphase and Telophase. Let's take up interphase. Interphase was formerly known as the resting phase, but actually the cell is highly active and prepares itself for the cell division. It is now known as the preparatory phase. The nucleus and cytoplasm remain metabolically active, and there is an increase in both nuclear and cytoplasmic content. The interphase is the longest phase and can be further subdivided into three phases: G1 phase, S phase and G2 phase. G1 phase is the first growth phase, also called post-mitotic gap phase. In this phase, synthesis of substrates and enzymes necessary for DNA synthesis occurs. DNA stands for deoxyribonucleic acid. Transcription of different types of RNA takes place. They are ribosomal RNA, transfer RNA and messenger RNA. RNA stands for ribonucleic acid. In G1 phase, synthesis of different types of proteins also occur. No change occurs in the DNA content of the cell. Sometimes the cell may withdraw from cell cycle and enter in the G0 or quiescent stage. Next is the S phase, also known as synthetic phase. Replication and duplication of DNA takes place during this phase. Entire genetic material, that is, the DNA, duplicates. Nucleus becomes double in size. Histone proteins required for organization of chromosomes are also produced. G2 phase is the second growth phase, also known as pre-mitotic gap phase. All metabolic activities take place, like synthesis of tubulin, the spindle protein, synthesis of RNA, duplication of cell organelles such as mitochondria and chloroplast, duplication of centriole forming two centrosomes. Thus, the cell prepares to enter mitotic phase. Thus, during interphase, the following events occur. The nuclear envelope remains intact. The chromosome occurs in the form of diffused long coiled chromatin fibers. The DNA amount becomes double. The size of the nucleolus increases due to the accumulation of ribosomal RNA and proteins. A daughter centriole originates. By the way, if you're getting some value out of this lecture, please like and share the video so that all of us can learn, unlearn and relearn together. Also, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Now we shall move on to the mitotic phase. The term mitosis was coined by Bolter Fleming, who for the first time observed dividing cells in 1879. Mitosis or somatic cell division is also known as equational division, since it maintains the chromosomal number of daughter cells similar to that of the mother cell. Mitosis consists of two main stages: karyokinesis or duplication of the nucleus. As shown in the slide, karyokinesis can be further divided into prophase, metaphase, anaphase, and telophase. Cytokinesis or division of the cytoplasm is the next stage. We shall discuss the phases one by one. Prophase is the longest phase. It can be divided into early prophase and late prophase. In early prophase, the animal cell becomes rounded, cytoplasm becomes viscous, the duplicated centriole lie close to the nucleus, the nuclear membrane and nucleolus are visible, the chromosome appear as long, thin threads in the nucleus. In late prophase, the nuclear envelope and nucleolus start disintegrating. The chromosomes shorten and thicken and their chromatids uncoil. Both chromatids remain connected with each other by the centromere, and both remain closely associated along their entire length. Each centriole separates and migrates towards the opposite poles of the cell. Spindle formation starts taking place from the centrioles. Next phase is the metaphase. The nuclear envelope and the nucleolus completely disintegrates. The chromosomes, as shown in the diagram, move towards the equator and get organized along the equatorial plane of the cell. Some of the fibers of the spindle attach with the centromere of each chromosome and are known as chromosomal fibers. Some of the fibers of the spindle remain attached from one end to the other end with the centrioles and are known as continuous fibers. Some fibers occur in between the chromosomes and are known as interzonal or interchromosomal fibers. The process of drawing the chromosomes onto the equator of the spindle is known as congression. So, we have studied prophase and metaphase. Next phase is the anaphase. It is the shortest phase of karyokinesis. The centromere of each chromosome divides into two. The chromatids of each chromosome are separated, very clearly shown in the diagram. They form two chromosomes. The chromosomes become shorter and thicker and migrate towards the opposite poles of the cell. Contraction of chromosomal fibers and stretching of interchromosomal fibers help in the movement of the chromosomes. The interzonal fibers push the daughter chromosomes towards the opposite poles. Next is the telophase, which is the final stage of mitosis. In telophase, at the poles, daughter chromosomes uncoil to form chromatin network. The nucleolus and nuclear envelope reappear. The mitotic spindles disappear. Therefore, after the telophase, two daughter nuclei are formed due to karyokinesis. This is followed by cytokinesis. The division of cytoplasm of the mother cell is called cytokinesis. Look at the diagram carefully. In animal cells, cytokinesis occurs by furrowing of plasma membrane that deepens and the daughter cells are formed. This process is called cleavage. Whereas in plant cells, it takes place by formation and extension of cell plate from center to the periphery. This process is clearly shown in the diagram. Finally, we will look into the significance of mitosis. Mitosis helps the cell in maintaining its proper size. A fully grown cell divides by mitosis. It provides the opportunity for the growth and development to organs and the body of organisms. Mitosis maintains chromosome number of parent cell in the daughter cell. It ensures equal distribution of genetic material and therefore maintains the characteristic of species. Mitosis helps in healing and repairing of multicellular organisms by forming new somatic cells. The old, decaying and dead cells are replaced by new cells. Mitosis helps the organism in asexual reproduction. Having studied mitosis in detail, we will now learn about meiosis. Edward von Beneden demonstrated reduction division in 1887. The term meiosis was introduced by J.B. Farm and Moore in 1905. Meiosis is a process in which the chromosome replicates only once but cell divides twice. It results in the formation of 4 haploid cells, each having half the number of chromosomes and half of the nuclear DNA amount present in the parent cell. Meiosis is therefore also known as reduction division. The meiotic division involves 2 successive divisions, Meiosis I or reduction division and Meiosis II or equational division, both of which have karyokinesis and cytokinesis phase. In first meiotic division or Meiosis I, reduction of chromosome number takes place and thus 2 haploid cells are formed. It is therefore known as heterotypic division. This division consists of 4 phases, namely Prophase I, Metaphase I, Anaphase I and Telophase I. Prophase I is the longest stage of the meiotic division. Its subphases are Leptotene, Zygotene, Pachytene, Diplotene and Diakinesis. In second meiotic division or Meiosis II, the haploid cell divides mitotically and results into 4 haploid cells. It is therefore known as homotypic division. The 4 stages of this division are Prophase II, Metaphase II, Anaphase II and Telophase II. Let us discuss the phase in proper sequence. First we come to Prophase I. It is the longest phase of meiotic division. Its first substage is Leptotene or Leptonema. The chromatin network condenses and becomes long and thin thread-like chromosomes in Leptotene. Each chromosome consists of 2 chromatids. The centrioles, already duplicated, move apart in pairs. There are 2 sets of chromosomes in a diploid cell undergoing meiosis, one set from the male parent and the other from the female parent. There are 2 similar chromosomes called homologous chromosomes. Next is zygotene or zygonema, in which homologous chromosomes begin to pair lengthwise, and this pairing is known as synapsis. A pair of homologous chromosomes lying together is called a bivalent. Next phase is Pachytene or pachynemia. During this phase, condensation of chromosomes progress and they become short and thick. The chromatids of each synapsed chromosome slightly separate and is known as dyad. The bivalents now appear to be composed of 4 chromatids known as tetrad. The 2 chromatids of the same chromosome are called sister chromatids, and those of 2 homologous chromosomes are called non-sister chromatids. Crossing over or exchange of genetic material between non-sister chromatids takes place. The points where crossing over takes place are called chiasmata. Next phase is Diplotene. The homologous chromosomes now start repelling each other and begin to separate. This is called disjunction. Separation of homologous chromosome does not occur at chiasmata. Nucleolus and nuclear envelope start disappearing. Finally, we come to Diakinesis. Here, chromosomes continue to condense and shorten. The chiasmata disappear by shifting to the ends. This process is called terminalization. The nucleolus and nuclear envelope disappear. Spindle fiber becomes prominent. Having studied Prophase I in detail, we shall now go to the next phase, that is Metaphase I. The spindle formation is completed. The tetrads move to the equator of the spindle and arrange themselves at the equatorial plane. Chromosomal fibers extend from the poles to the centromeres of corresponding homologs. Next, we come to Anaphase I. In this phase, from each tetrad, 2 chromatids of a chromosome move as a unit to one pole, and the remaining 2 chromatids migrate to the opposite pole. At the end of Anaphase I, half the number of chromosomes move to respective poles. Next is Telophase I. The chromosomes uncoil and elongate. The spindle fibers disappear. The nucleolus, nuclear envelope, reappear. After karyokinesis, It is division of the cytoplasm, that is cytokinesis. The cytoplasm divides by cleavage or furrowing or by constriction in an animal cell and by cell plate formation in a plant cell. This produces two daughter cells, each with one nucleus. A very short resting period between Meiosis I and Meiosis II is called Interkinesis. It may or may not occur. Next is the second meiotic division or Meiosis II. First comes Prophase II. Prophase II of meiosis shows the chromosomes with two chromatids which become short and thick. Centrioles migrate to the opposite poles. Nucleolus and nuclear membrane disappear. Prophase II is followed by Metaphase II. The chromosomes get arranged on the equator of the spindle. The chromatids of each chromosome are joined at their centromere by spindle fibers, namely chromosomal fibers. Next phase is Anaphase II. In Anaphase II, the two chromatids of each chromosome start moving away from each other. Each pole has haploid number of chromosomes and haploid amount of DNA. This is followed by Telophase II. The daughter chromosomes start uncoiling. Chromosomes are enclosed by nuclear envelope. Nucleolus reappears. In cytokinesis 2, cytoplasm in animal cell divide in the middle by furrowing and in plant cell by cell plate formation. 4 haploid daughter cells are formed at the end of Meiosis II. Now we come to significance of meiosis. Maintenance of chromosome number. Meiosis maintains a definite and constant number of chromosomes in the organisms. It produces haploid gametes for sexual reproduction. It introduces genetic recombination, leading to variations and evolution. Meiosis produces spores in plants for asexual reproduction. We have covered mitosis and meiosis in detail. Now we will look into the differences between mitosis and meiosis. Mitosis occurs in somatic cells. Meiosis occurs in germ cells only. Mitosis shows only single division resulting in 2 daughter cells. Meiosis involves 2 successive divisions resulting in 4 daughter cells. In mitosis, the prophase is of short duration. Substages are absent. In meiosis, the prophase is of longer duration and comprises of Leptotene, Zygotene, Pachytene, Diplotene and Diakinesis. Pairing or synapsis is absent in mitosis. Pairing occurs between the homologous chromosomes in meiosis. Chiasma formation or crossing over is absent in mitosis. Chiasma formation or crossing over takes place in meiosis. No exchange of genetic material in mitosis. In meiosis, exchange of genetic material takes place between the non-sister chromatids of homologous chromosomes. In mitosis, anaphase involves separation of chromatids of each chromosome. In Anaphase I of meiosis, separation of homologous chromosome take place, chromatids move apart in Anaphase II. Telophase always occurs in mitosis. Telophase I is eliminated in some cases during meiosis. Mitosis brings about growth, repair, and healing. Meiosis forms gametes or spores and helps to maintain constant number of chromosomes. So today we have learnt about cell cycle, cell division, interphase, mitosis and meiosis in detail. I have some practice questions for you. Please share your answers in the comment section below. You can pause the video if you like and write down the questions. Thank you so much for your time and participation. If you haven't subscribed to my channel yet, please consider subscribing the channel and press the bell icon to get instant notification of all the upcoming videos. Also, if you want to discuss any particular topic in biology, please mention that in the comment section below. I'll see you there. Goodbye. All the best.